All right, everyone, it's 12:02, so we'll go ahead and get started. My name is Katie Gertis. I'm the Director of Alumni Relations within Health Sciences at the University of Pittsburgh. Thank you for joining today's Pitt Health Sciences Virtual Health Academy. Today, you will be hearing from Ken Gabriel on the what and how of BioForge. I wanted to offer a quick reminder before we start with our formal presentation today. We will open the floor for Q&A after the presentation. So if you have any questions you would like to ask, please utilize the Q&A feature that is available within the platform, and we will address as many questions as we can. I would also like to take this moment to formally introduce Ken Gabriel. He is the inaugural Chief Executive Officer of BioForge, an organization using $100 million in seed funding to accelerate breakthroughs in the development and manufacturing of biological precision medicines to speed their impact. He also sits on the board of companies in a variety of industries, including Trimble, Galvani, and Dover Microsystems. Throughout his career, Ken has delivered game-changing innovations to the private sector, government, and academia. Ken earned a Bachelor of Science degree in electrical engineering from the Swanson School of Engineering here at the University of Pittsburgh, and a doctorate in electrical engineering and computer science from the Massachusetts Institute of Technology. It is a pleasure to have Ken here with us today, and I am happy to pass the program over.
Thank you, Katie. All right. Welcome, everyone, even though I can't see you. So, excuse me a second. You were going on here. Apologies. I need to share. Even though we worked this out. Now, welcome everybody. It's a pleasure to be with you, even though I can't see you. As Katie introduced, I am here to give you a quick overview of BioForge, and in particular, the what and how of BioForge. So the what is pretty straightforward, and one that we can wrap up and describe in this statement. We are focused on creating innovations and breakthroughs in the manufacture of precision biologic medicines to speed their delivery, use, and impact. Now, I'm sure, like you and I, you have seen various articles come out over the past several years about the promise of precision biological medicines. The upper left was about a year ago. UK toddlers' hearing was restored in the first gene therapy trial in the UK. The article to the upper right that's larger is a more recent one from earlier this year. Again, a baby with a rare disease was cured completely by given a personal CRISPR gene therapy. You can't help but be touched by these sort of developments. These pull at the heartstrings, the toddlers that can hear their parents for the first time or live to have a productive life. The challenge, though, is often as you read these articles, a little bit later comes the not so good news, which is that these things are super expensive, anywhere from $300,000 to a single treatment or $4.5 million per treatment. Clearly, this is not something that is going to have an impact and be broadly accessible and available unless we tackle what is the challenge in delivering these precision medicines. And certainly we need to find, as this market study pointed out, better ways and less expensive ways to make these products available. So that is the what. We are focusing on how to create breakthroughs and innovations that makes them more affordable and accessible to a greater number of people who need them. Now, to give you a sense of what it means when these treatments are so expensive, let's take a look at the scale of what this means. Since 2017, when the first of these gene therapies that are labeled CAR T-cell therapies, there have been slightly more than 30,000 leukemia and lymphoma patients that have received that first sort of genetic treatment for a cancer. That's an average in the last 10 years of about, or 8 years, of about 4,300 patients per year. Well, in 2024 alone, the American Cancer Society estimated that there was 143,000 new cases and 43,000 deaths, the most severe of those cases. What that 4,300 patients, just to give you a sense of what this cost is doing, we're treating only 10% of the most advanced cases per year. That's what these costs translate to. A small fraction of the people who need these sort of treatments. We think that it's not enough to just not only treat so few people, but to constantly be saying, well, eventually the price will come down. The price is not going to come down, or the costs aren't going to come down until there's direct and deliberate focus on the manufacture of these precision biological medicine. So, as Katie pointed out in the beginning, the BioForge was kicked off with the largest single grant from the Richard King Mellon Foundation to the University of Pittsburgh back in 2021, 2 years before I got here. And that was matched by 120 million provided by Pitt for the core and shell of a new 185,000 square foot building in Hazelwood Green. So when I came on board a little over a year and a half ago, one of the things that was important for BioForge to be able to do what it needed to do in this new era of creating manufacturing breakthroughs was to be an independent organization that could move with the speed and agility that was needed to create those breakthroughs and innovations. Now, while we're an independent organization, we are a wholly owned subsidiary of the University of Pittsburgh. Where building is going up in the Hazelwood area, right across the street from the Carnegie Mellon's University Manufacturing Futures Institute. So it's an interesting neighborhood to be in. The old Mill 19 that you see up there in the upper left, an old steel building, steel mill, one of the last ones, actually the last one to shut down in 1996 in the area. So part of what's happening, part of the investment from the Richard King Mellon Foundation, from the University of Pittsburgh, is creating, shifting Western Pennsylvania from steel to cell manufacturing. Now, this shift is also creating an opportunity. Anytime you have shifts in what are treatments, what are therapies, they create opportunities anytime there's technology shifts. And what is the shift happening in pharmaceuticals because of precision medicine? Well, it's an inflection in the type of pharmaceuticals. So what you see on the left here is the market in billions of US dollars for pharmaceuticals globally. And what is the big thing is that there's two types of pharmaceuticals, as you can see here. Chemicals, small molecule chemicals like aspirin and other drugs that we are used to taking and have been predominantly the therapeutic, and so-called biologics, things like Humira and other treatments that you may be familiar with that are not small molecule chemicals, but are produced by biological and are biological products. not only are the biologics increasing in general, but the precision biologics are creating a different sort of demand. So as we move from chemical therapeutics, chemical drugs, to biologics, that's one shift. The other shift is that when we go to precision medicines, things that are designed around genetics and personal genetical makeup, that's creating a different type of shift and pressure on pharma manufacturing equipment and tools. So today, obviously, I pointed out the one, we're moving from small molecule chemicals to biologics. But because of the precision medicine element, we no longer can produce huge volumes in batch fabricated procedures where you make 1 billion of the same therapy, because precision by definition means you're developing therapies, manufacturing therapies that are in smaller volumes, not billions, maybe hundreds of millions or tens of millions, or in the ultimate case with some of the specific CRISPR and other techniques, just an N of one, just one patient. So that means we have to go from high volume batch to new types of equipment that produces low volume and continuous fabrication. We have to shift from things that take months to reconfigure and change from one type of treatment to another and cost millions, to hours for reconfiguration and change and less than $1,000. And then finally, the really important part, which is why this region, why Pittsburgh region has a right to win in this area, we're moving from touch labor and open environment and traditional industrial control to automated inline monitoring, machine learning, and AI control. You're only going to get these rapidly reconfigurable systems and low volume continuous fabrication with systems that have inline sensors, controls, and facilities. So this is not simply, what I want to reiterate, it's not simply moving from chemicals to biologics, but a complete redo of the pharma manufacturing industry ecosystem, equipment, and tools. So that's the what of BioForge. What's the how of BioForge? It's equally important that we innovate in the way that we approach this problem. The what can be really revolutionary, go after breakthroughs and innovations, but if we use the same old approaches, we're going to have the same old answers. So what's really different about BioForge is there are numbers of multiple projects that will be going on inside of BioForge, but with no large established organization. So unlike either in the private sector or in the academic sector, BioForge permanent employees or core employees will never be more than about 15 or 20. But at any one time, we may have 250 people working at BioForge. The way that works is we will have, as this chart is trying to show and demonstrate, at any one time, we'll have a number of different projects that are ongoing. Some, like at the top, that are finishing up or have finished, some in the middle that are roughly midway through their activities, and others that are just beginning or being considered for a project. So each project within BioForge there will be multiple diverse projects that BioForge will host, resource, and support. Each project is a small group of people, 5 to 10, with diverse backgrounds in biology, life sciences, and engineering and physical sciences, working together over fixed duration, fixed budget projects, typically from 18 to 36 months tops, all focused on demonstrating that breakthrough and innovation in the manufacturing. Where we will get those ideas? Well, the University of Pittsburgh Schools of Health Sciences are generating discovery-driven results every year. depending on how you count the research results and what's happening in Washington, but upwards of $800 million a year in R&D funding that's coming out of the schools of health sciences. From those, some of those research results will have broad impact, have the potential for broad impact, are limited by manufacturing, and we believe there's something we can do about it. So those activities, some of those research results will get pulled in as a project, and the selected research projects will have skin in the game from the people who did those discoveries. What I mean by that, while BioForge will do the bulk of the resourcing and supporting of the projects, we expect to have in-kind people who came up with those discovery-driven innovations to be part of those teams and working towards that demonstration at convincing scale. Now, what are the possible outcomes from the how of BioForge, these diverse multiple projects that are both time-bound and budget-bound? Well, they can either lead to a convincing demonstration that leads to funding and a spin out of a startup, or something that may be successful, the demonstration is convincing, but the technology is more suitable for a large pharmaceutical company, in which case we would license that IP and capabilities. But it's also important to realize that one of the ways we're structuring this is to have a bold reach. And if our reach is bold enough, some of these projects will give a good effort, but there won't be a successful convincing demonstration at the end. That's okay. We clear the decks because we need to bring in new projects and give them a shot at the opportunity inside of BioForge. This is not, like your typical research center that's going to sit and work on a particular project for 10 or 15 years. This is one where there's going to be a vibrant churn of new projects coming in, because in those 18 to 36 months that you did to experience, there's new discovery-driven research results coming that deserve a shot at a slot at BioForge. So, excuse me, the building's going to be finished. The core and shell is up. There's going to be another year to year and a half of fit out and additional equipment inside and facilities and office, but we're not waiting until that building is finished. And for the last year and a half, we've actually been moving into several different temporary interim spaces here down at Bridgeside Point, if you're familiar, near the hot metal bridge. There's multiple buildings and facilities, and we're moving into some interim space to get projects going. We have started and have underway 3 projects in the last year and a half. One that is working on creating access and democratizing the access to mRNA fabrication to increase the number and diversity of designers. We've launched that activity, and we now have a running brokerage service that was designed to go global later this year. We have a second project that got started in dissolvable microneedle arrays, which is a precision skin cancer delivery, not from a genetic precision perspective, but from a precision dosage, location, and delivery of a particular cancer therapeutic. That project is actually the first of our projects to be finishing up. And in 10 months, we kicked this off roughly last year, 10 months ago. The project has closed out with Panther Life Sciences, the startup that had licensed the technology from the dermatology department, having 8 employees in Pittsburgh over the last 8 months established, and with a decision to establish their manufacturing and materials site here in Pittsburgh, targeting 20 plus additional employees over the next few years. So this is a bit of a hybrid in the outcome that I talked about. It's both a startup plus a licensing opportunity to set up operations here. The third project, which we just got started about 5 months ago, is looking at an area called adeno-associated virus delivery. This is a traditional 30-year-old technique to deliver genetic treatments, but that has had rough time with the manufacturing, has fallen out of favor. We're looking at it to see if we can create a general delivery platform that could get around many of the limitations that have kept this technology from being used. In the pipeline is some additional stuff, including peptide cancer vaccine cocktails that we'll talk about some more, perhaps maybe next year. But what do I mean by BioForge resources and tools? We are not only creating a traditional laboratory for biological systems development and manufacturing, but we're bringing the best in class and sometimes just coming out technologies in nano 3D printing. This Nano 1 Green, which got delivered and certified 2 months ago here, is creating structures. It's 3D printing, but at a scale where we can resolve at 100 angstroms of resolution in the z-axis. Why is that important? When you're creating new ways of getting cells and cellular biochemical reactions and biological reactions to happen, you need to have the reaction chambers and the reaction structures at the same scale as the cells and some of the proteins. So this is the type of technology innovation that creates the opportunity to do things in a completely different way that was not available as recently as just 3 years ago. So these are the fusion of biological processes, life sciences, with physical and engineering approaches that are going to create these innovations that we've not had access to before. The example of another resource and tool we're bringing in, which has more of a biological flavor, is the ability to take a state-of-the-art machine. This is called the DNA script syntax machine. This is a company in Paris that's been financed by Illumina Ventures to create cell-free manufacturing of DNA through a collection of oligonucleotides being attached together. So the pieces of the DNA are made in a traditional chemical synthesis, enzymatic, not inside of a cell. And then a second machine that you see down below, a fluidic handling system that puts them together to form the long and specific DNA construct that is needed for the either generating RNA or direct integration into cells for genetic treatment. As I said, the team is relatively small. So far, we are up to 8. We expect to stay that way for at least another year. But at any one time now, we have an additional 15 people working on those 3 projects that I talked about. Finally, I mentioned the building that is being built. There's the architect's rendition in the upper right-hand corner. You see where that building is relative to the Almano site, the Allegheny Monongahela site that is down there by Hazelwood Green, the site of the Mill 19 building. And one of the things that I want to point out is as part of this construction, there's going to be 185,000 square feet. And we already have, through Pitt's efforts early on, an anchor tenant with a company out of Waltham, Massachusetts called Elevate Bio, who will be setting up their second manufacturing site for cell and gene therapy production for actual use by patients in the building. So they'll have about 70% of the building, an actual, contract research and manufacturing organization. embedded here in Pittsburgh, and BioForge will move out of our interim space and will take about 30% of that 185,000. The building, as you would see it from the riverside, is here again in an architectural drawing, but this is just about a month ago. This is the actual building. It's taking shape. And as I said, this is the shell and core with the interior fit out coming a little bit later in the next year to 16 months. This is a picture right at dawn, which I thought was appropriate because we are literally at the dawn of precision biological medicines, which is going to change the way we not only treat certain diseases, what kind of therapeutics we come up with, but that are going to demand a completely different approach to the manufacturing and the associated tools and ecosystem. So that's my quick what and how of BioForge, and I'll turn it back to Katie to see what sort of questions have popped up over the course of the presentation.
Great, thanks so much, Ken. We did have a few questions come through.
Yeah.
So our first question would be, what are your priorities in the behavioral health space?
In the behavioral health space,
Mhm.
there are, so one of the things, the approach that I've taken to BioForge is to avoid us being the ones who define what the priorities are. So I spend 40 to 50% of my time talking to all different parts of the university, particularly within the health sciences. And it's from those interactions that these projects have come in. And so today, we don't have any projects per se in the behavioral sciences space, but I can tell you there are emerging developments in discovery-driven research where there are certain biomolecules, nutrients, and other things that happen at the blood-brain barrier that are coming out of Suzanne Amari's work in the psychology department, where there may be, I would even say, a pre-candidate ideas about how precision nutrition or precision targeting of certain types of neurotransmitters might have an impact on behavioral sciences. But I want to emphasize, I don't consider my job to be to define specific areas for BioForge to go into, but rather to describe what is BioForge capable of doing and who out there with the different discovery-driven research needs or could benefit from us helping with certain manufacturing or delivery and scale challenges that their discovery-driven researchers needs.
Cool, thank you. Another question, does your model include a scenario where a Pitt faculty brings on board two existing companies, one of them which licensed Pitt cell therapy IP, and the other who can provide unique manufacturing resource to reduce the costs and allow broad application of the cell therapy platform across multiple academic and non-academic users?
Yes, if I understood that question correctly, one of the things that, as I mentioned in the how, we expect some skin in the game from the folks who developed, as a result of the discovery-driven research, who developed the demonstrations and the science that needs to be scaled up and manufactured. And one of my principal tenets as part of the how is I don't want to reinvent things that already exist. So if a faculty member is coming with an innovation and they already have, say, in years prior, done a startup, and it's a company that's commercializing their work, and the faculty wants to bring that in to the project, by all means, because I don't, the biggest threat to innovation and breakthroughs is time. So that's why you see us being very aggressive about timescales, but also I don't want to waste time or resources on things that already exist. I don't need to reinvent them. So if a faculty member is coming and they're coming in with a startup that they're either started or partnered with, by all means, what we'll build them into the project so that we can get to that demonstration of the manufacturing breakthrough as quickly as possible.
Great, thank you. Another question that came in was, with the current range of costs for gene and cell therapies being between 300,000 and over 4 million per therapy, how much of this cost is related to the related manufacturing processes BioForge is challenging, and by how much do you think costs could be decreased?
Yeah, so that's a great question, and one that I'm glad to address, because if you look at the material cost when the actual therapy is developed, the material cost certainly doesn't justify the 300,000 or the 4.5 million. If you look at the processing, yeah, there's a little bit there, it's complex and it's not particularly efficient or best-in-class manufacturing approaches. There are a lot of factors that influence the ultimate cost. And in fact, when people talk about the cost, it's not like you can go and buy it yourself. These children and adults who get these treatments are brought in as part of a clinical trial. So I recognize that there are multiple factors, including regulatory factors, clinical trials that the pharma companies have to extract the cost of from the ultimate therapies that they give. But my approach, and one that I believe will be successful because you've seen it be successful in other areas, is I believe we can drive the costs down by a factor of 10 or 20 at least, and get these treatments, which are $1 million or more, down to $25,000 to $50,000. When you do that, I think if we're able to do that, then you'll have patient advocacy groups that will push on the medical system. I can't fix the entire medical system. If I tried to do that, everything would collapse. I'd be here for 100 years, nothing would move. What I can do is make the actual cost of the therapies be very clearly understood, lower by a factor of 10 or 20, and then that will put pressure on the rest of the system to make it available more broadly and equitably. But I think we're talking at least one order of magnitude potential price reduction, and possibly more than that. But definitely at least a factor of 10 or 20 from everything that I've seen.
Cool, thank you. Next question. Do you anticipate hiring of the people who work on the specific projects to expand? And if so, how do young researchers learn of these employment opportunities?
So we do hire, I expect right now in our interim spaces, we will probably have room for maybe 5, and if we push it, maybe 6 projects before we can move into our final BioForge building in a year and a half or so. Each project is going to require a different number of people, some more than others, and a different diversity of people. We will bring in people, hire them for the projects, but they will be term hires for those projects, somewhere between 12 to 36 months. The way we typically advertise for those positions, as we have for these 3 projects we have on hand, has been through LinkedIn. and we post on the University of Pittsburgh site. We're working with them to give us that advantage, but by the same token, we want people to recognize that they're not being hired as Pitt employees if they come to BioForge. Because remember, BioForge is a completely independent organization. We're an LLC, but we're wholly owned by Pitt.
Great. Thank you. Our next question is, with this spinoff and IP opportunities, is there any financial relationships with banking and financial firms that can help connect capital, people with these ideas coming out of BioForge to really help make successful launches?
Yeah, so we're early in the process. Panther Life Sciences, the one startup that I shared with dermatology, had already secured long before they came in as a project partner. They had already secured the licenses from Pitt. They were financed by both the founder and others that had put money in. I think that is going to be certainly, that's an important element. But one of the things I've learned throughout my career, and I've done startups, I've done publicly traded companies, I've done large research organizations. It's really important to make those opportunities clear to people who have the money. So whether it's banks or venture capital or others, but to bring them into the process tends to can make both the funding and the research get a little bit out of whack. So it's important to finish the research, and then we're happy to form relationships with banks and venture capital, but not until those entities are ready to go out, because it's really important to separate and keep people focused on demonstrating the breakthrough, which is a very different set of tasks and deliverables than what you need when you start getting venture funding and you have a company and you have a minimally viable product you need to produce. So we're open to it. We're early in the process. And this is one of those areas where we think it'll be important for the visibility of our projects when they're ready to show that the region is generating opportunities for investment.
Great. Thank you so much. So that does wrap up our Q&A for today. We really appreciate your time, Ken. Thank you for sharing your knowledge on BioForge. I hope you all enjoyed the program. You are free to go. If you have any questions, feel free to contact me. Kyla will put up a slide with some information, and there will also be a recording available within the next business day or so that will go out to all who registered if you would like to rewatch the program. Everyone have a great afternoon, and thank you again, Ken.
Pleasure. Take care. Bye-bye.
Bye.